This presentation is titled Regenerative Medicine: From Signal to Structure. This work is presented by Brian Geniuson, founder, principal research scientist, and research systems architect at Natural Scientifics. The goal of this presentation is to explore regenerative medicine from a systems biology perspective, examining how molecular signals, cellular behavior, and tissue architecture interact to determine whether injury resolves through functional regeneration or maladaptive repair. This content is provided for educational and scientific discussion purposes only. The extracellular matrix is often described as structural support, a scaffold that holds tissue together. But biologically, the extracellular matrix is not passive, it is not background, and it is not simply something cells attach to. The extracellular matrix is a control system. In living biological systems, tissue healing does not depend solely on the presence of regenerative cells or molecular signals. It depends on how those signals are interpreted by structure. It depends on the physical properties of the surrounding matrix, and it depends on whether the tissue environment permits regeneration or enforces repair. Healing outcomes emerge not from signals alone, but from the interaction between signal and structure. At the center of this process is the extracellular matrix and tissue microenvironment. Matrix composition, collagen organization, mechanical stiffness, inflammatory signaling, and vascular perfusion collectively shape cellular behavior. When these variables remain within physiological bounds, progenitor cells retain flexibility and regenerative programs can proceed. When they fall outside those bounds, the same cells are driven toward fibrotic repair rather than tissue renewal. These factors do not act independently. They amplify one another. Increased matrix stiffness alters mechanotransductive signaling. Altered signaling promotes fibroblast activation. Fibroblast activation accelerates collagen deposition and cross-linking. Inflammation reinforces these changes, while reduced perfusion limits resolution. Over time, this feedback loop locks tissue into a repair-dominant state. Every moment following injury, tissue exists at a decision point. When matrix elasticity, inflammatory timing, and perfusion are balanced, regeneration can restore structure and function. When imbalance persists, repair preserves continuity at the cost of architecture. The extracellular matrix functions as a gate, not permitting regeneration unless structural conditions allow it. This is why regenerative therapies often fail despite promising cellular or molecular components. The failure is not always the signal. It is frequently the structure. Cells introduced into a fibrotic or mechanically hostile environment cannot execute regenerative programs, regardless of their intrinsic potential. Regeneration is constrained not by intent, but by context. If regeneration is governed by the extracellular matrix, then regenerative medicine must engage structure as deliberately as it engages cells and signals, not as an afterthought, not as passive support, but as a primary determinant of biological outcome. In future discussions, we will examine how this decision gate operates across specific tissues, why fibrosis represents a locked structural state, and how regenerative capacity intersects with aging, injury, and disease. A full scientific breakdown of the mechanisms discussed here, including matrix signaling pathways and supporting literature, is available separately. This presentation is for educational purposes only and does not provide medical advice.